Welcome to Innovate and React. I'm super excited about this episode because it will be about a variation of my favorite analytical method, nuclear magnetic resonance spectroscopy. So I'm very happy to welcome Anne Meyer to the show. She's using solid-state NMR for protein research in her PhD. Hi, Anne.
Yeah, hey, thank you for your invitation.
Let's start with the beginning. How did you end up in this field? I think solid-state NMR is not that common.
Yeah, that's true. So maybe I start with my beginnings with my bachelor. So I did chemical biology during my bachelor, and then I decided that I want to do more physical chemistry and to focus more on NMR and theoretical chemistry. And so I ended up in Frankfurt, which is a huge university for membrane proteins. And we have a lot of methods how we investigate them. And one main method in Frankfurt is NMR.
Mm-hmm.
So we have a lot of spectrometers. We have this BMRZ, which is called Biomolecule Magnetic Resonance Center. And yeah, it's huge. And during the master, I did like every kind of magnetic resonance in general. So solid state NMR, liquid, or I always say liquid NMR because solid, liquid, no, but it's solution NMR. And EPR, which is electron paramagnetic resonance. So it's similar. And after my thesis, which I actually did not in NMR, I did some theoretical chemistry, but I missed NMR and magnetic resonance. So I, more or less decided like which internship I liked the most. And this was in the solid state NMR group. So here I am.
Yeah. We will then look into how this works later. It's like we also in Dortmund had a quite big NMR department, and that's how I got interested into NMR. And this started, or the interest really sparked when I went to the lecture for the NMR, and the professor talked about how this whole method came about, and that it is the first technology that has a Nobel Prize in all three scientific fields. So it's a fun fact that I think is really interesting. In total, there are actually 5 Nobel Prizes for the discovery of NMR and its applications. The first 2 were in physics. In 1944, Rabi was awarded the Nobel Prize for the discovery of NMR. And in 1952, the discovery of precision measurements laid the foundation for NMR. And about 40 years later, Ernst got the Nobel Prize in chemistry for the first time then, for high-resolution NMR and adding the Fourier transformation NMR, which made NMR usable for analyzing molecules. And then in 2002, NMR got the prize for determining the 3D structures of biological molecules in solution. So that's also close what you do, just not as a solid form. And finally, in 2003, Lauterbauer and Mansfield got the Nobel Prize in Medicine for the discovery of magnetic resonance imaging, so MRI, which is used today a lot in medicine to basically look into the body without having to cut it open. And that's pretty amazing. Now let's dive into how this all works. Let's start with the basics.
Yeah, okay. So maybe like NMR, just to repeat it. So it's nuclear magnetic resonance, and I maybe think it's useful if we go word by word through the things. So nuclear means like that we do something with the nucleus. What we actually do is to excite the proton spin, in the nucleus. And we do that in NMR with radio frequencies. This is a method where you use radio frequencies to excite the proton spin of a suitable atom. And when you look into your periodic system of elements, you can see a lot of elements, but which are the suitable ones? This is defined by the number of neutrons in your nucleus. So, for example, we have what we use commonly in bio NMR. We have the hydrogen, which has 0 neutrons in the most common form. And then we have 13 carbon and 15 nitrogen.
These are the most common, right? To measure for chemists in the lab, usually you go for hydrogen and carbon, I would say. Nitrogen is more for biology, right?
Yeah, exactly. So there are a lot of other NMR active nuclei. So you could also measure sodium. But these ones are the most common ones and the easiest ones because you excite the proton spin. But there's a next thing you should consider, how the magnetic spin is divided after you put it into the magnet. So for protons, that's how you call hydrogens in NMR. They are not hydrogen anymore. So for them and for the nitrogen and also the carbon, you after you put the sample in your magnet, then you have the external magnetic field, which is called B0. That's important.
That's the magnetic part in NMR.
Yeah, thank you for noticing. Yes, exactly. So now we know, okay, nucleus, protons, neutrons are important. So now we go further to magnetic. So so we have this external magnetic field and B0. And when you put your sample into the magnet, then something happens which is called Zeeman distribution. So for example, for hydrogen, 13 carbon or 15 nitrogen, you have a splitting into 2 different energy levels. There are other NMR active nuclei which split into more energy levels, but this is the hard case. So I think I will just focus on the easiest way. So this has something to do with magnetic number and some quantum stuff. And so, but in this case, we have for hydrogen and the other easy ways to explain it, we have spin one half. And so we have alpha level, which is lower in its energy, and we have a beta level, which has a bit higher energy. And yeah, you can just imagine two bars in your head. So you have one bar in the lower and the other one is just a bit higher. And here you have all the spins in your sample. And because of the Boltzmann distribution, the population of the alpha spins is just slightly bigger than the one of the beta spins. And those few spins, we have more at the alpha level. These ones we can excite with another magnetic impulse. So here comes the radio frequency in, and here comes this resonance part. Because we have this radio frequency with a specific resonance frequency that you can really excite those. This frequency depends again on the spin you want to excite. So you have to use a different frequency for hydrogen or carbon and stuff.
Because the energy levels are different?
Yeah, they're split, they divide differently.
When you radiate the nuclei which are in the B0 field with the radio frequency, what happens then with the spin?
Yes, so the small amount of spins which can be excited changes its magnetization. So those come through, like in general, all changed magnetization, but these ones are the excited ones. And now they are excited. And when you imagine a coordinating system, so in the beginning, we have them along B0, which is like the z-axis, and then you excite them and they go into the XY plane. And in here we can detect them. So here are our detectors, and what they do now, if you do nothing, so you just excite them and then you stop the pulse. So this is just a short pulse. The B0 field is there all the time. It's constant, but the radio frequency pulse is just a short one. And then in the XY plane, you have all of your spins, and then you stop the pulse, and then they go back into their equilibrium. And during this process, we can detect them until they are again along B0.
So everything that is not aligned with B0 is measurable, basically?
Yeah,
Okay.
but in the end they are back.
Yeah, they always
Back
go
on
back
track.
to the strong magnetic field.
Yeah, so this is like the time they need to go back. It's called T1 relaxation time. Yeah. So depending on the spins and also the sample and a lot of other things, they need more or less time to go back. Exactly. We also have another relaxation, which is called T2. So all these two relaxation influences, they define how fast the spins relaxate back to the along B0. And what we measure is this is called FID. And the FID, now here comes also again the Fourier transformation. This is a frequency spectrum, and we Fourier transform it to signals that we can read out. And depending on all of these influences, the spins are affected during the time going back to B0. They have another chemical shift. And now one could ask, what is chemical shift? And yeah, that's the definition of how much things are shielded. Yes, so we have the spectrum then in the end with the chemical shift. If you have a strong interaction with surrounding atoms, the chemical shift is higher. And if you are if everything is shielded, then you have a lower chemical shift.
Shielded is with electrons?
No, more electrons in general mean higher chemical shift. So aromatic rings, they have a high chemical shift. No, shielded X means that there are methyl groups are highly shielded. So they are just like the hydrogens and the carbons, but not so many free electrons they can interact with. So this means shielded. So no, the lack of interaction, more or less.
Okay, yeah.
And the unit is ppm, and ppm means parts per million, and we use that as a clever trick because we have a lot of different magnets. So the B0 field is not the same for every spectrometer, but we want to compare the spectra with each other. And therefore, we use ppm as unit. So that's pretty useful.
So you can compare, for example, when you have a 1 Tesla magnet, then you can measure something. You will get your signals with the Fourier transform, and then you can read out the ppm. And this will also be the same when you measure the same molecule in a different spectrometer with a 2 Tesla magnet. I don't know. What's the range of magnets you use for NMR?
Yeah, the range is huge, but the ones we typically use are starting from 400 megahertz. So we usually don't use Tesla as unit in NMR. Tesla is more common in EPR, if I remember it correctly. And like one can say for sure Tesla, but we say megahertz. And so we're starting with 400, but we have a 1.2 gigahertz spectrometer in Frankfurt. But the ones we usually use are the ones with around 600 or 850. This is like something we usually use.
Quick break. I want to reach more chemists, more scientists, to share these discussions and hopefully spark new ideas. For that, I want to ask a small favor from you. Please hit the follow button. This really helps me to grow the show, and it doesn't cost you anything. Thank you for your time. Back to the episode. And the gigahertz and megahertz, they are derived from the proton frequency with this specific magnet, right?
Yeah.
So that's how you can convert from magnetic field. The higher the frequency, the stronger the magnetic field would be.
Yeah. So the resolution with higher magnets is better because the signals are more separated. So, and this is pretty useful for hydrogen because in general, all of the substances we investigate are with a lot of hydrogens. Yeah, this is a big advantage that you have a higher resolution. For solid state NMR, it also has some disadvantages. Maybe I can come back to this later because I think we should maybe start what the differences are.
Yeah.
Okay, I think like just to sum it up, so we have spins, proton spins in a B0 field and excite them with B1, which is our radio frequency. And we learn something about our sample because we detect what happens when it comes back to B0. So this is the important time.
The resonance, you basically listen to what the nuclei give back from this excitation you give with the B1, right?
That's nice. Yeah, we listen to them.
Also, to maybe shortly explain how really useful this Fourier transformation is, when you have all these molecules in your solution for now, then you would basically get a super noisy signal because you hear all the nuclei at once. They all are a bit different, but they would overlay. And with this Fourier transformation, you can get defined signals because each wave can be derived to one signal. I think more or less. I don't know how this, it's really complicated math, so I don't know exactly how this works, but basically it divides up the different waveforms you get, and then you get one signal for each atom.
Yeah, that's true. So it's really complicated. I did this once during my master, and it was wow.
You did it with like per hand, basically. You calculated everything.
No, in one exam we had to do actually Fourier transformation by hand with a specific function.
Oh, okay.
Yeah, this sucked. It was hard. It was pretty hard, but this whole exam was crazy.
Yeah.
Yeah, but that's true. So what we still see the spectrum, or it's not a spectrum, and just we see a curve when before we Fourier transform it. It just looks like a wave. So you can't see anything. And then we do the magic, and then we actually see like the things I explained with the chemical shift and
the signals.
Yeah, as very clever people with a lot of time and a lot of thirst for knowledge, they found out which substance is which chemical shift. So now that's the crazy thing. So now we know so many things about NMR.
Yeah.
But all these people who got the Nobel Prize, they were really crazy. So this is amazing.
Yeah.
And I really love the method. I hope I could explain it a bit, but just as like the Bible for the more interested people in NMR is definitely Spin Dynamics by Malcolm Levitt.
Yeah, I can link that in the show notes.
It's the NMR book from Keeler. So these two books are really nice if you want to understand more. It's a bit hard sometimes to explain it without pictures, but
And also it's all this physics stuff, the fundamentals are really complicated. And then when you apply all this knowledge that these people got us in the past, then it gets a bit easier. But as you said, it's a bit difficult to explain without images.
yes, that's true.
Okay, to quickly sum this up, we have a magnetic field B0, which aligns all the spins in the sample. Then we need NMR active nuclei to look at, which is the case when at least one of atomic number and mass number are odd. And then we get in this magnetic field a Zeeman distribution, which splits them into different energy levels and makes them excitable with a second magnetic field B1. This is usually a radio frequency. And with this radio frequency, we excite the nuclei and then we listen to the resonance of that, which is basically by the excitation. They get out of the alignment with B0 that we can measure, and then they basically go back to B0.
Yes, this is like the basic. This is how also MRI works. So protons, spins get excited. And in solution NMR, the dynamics are completely different compared to solid samples, or in my case, membrane protein samples, which are definitely not liquid. And so the thing is, in solution, everything averages out. So we have a lot of isotropic effects. We can see a lot of things, and we can see things we could also see in solids, but which are absolutely negligible during our measurements because we have other things to do.
In solution, they just move around so much that they are basically in all orientations when you average out all the molecules you have in your solution, and then you don't see these interactions, right? But when they're in a solid, they are confined and they cannot move, and then you see interactions that you would not see in solution, right?
Yeah, more or less. So we have like what we have in solution, everything is like a ball or like a sphere, and it's very round and very perfectly shaped. So in every, like, again, imagine the coordination system, everything is perfectly shaped in this. And what we have in solids, everything moves much slower. We don't have this perfectly shaped sphere. We have a tensor, which is elliptically shaped. So we have in one direction is more than in the other one. So we have this anisotropy. With this, we come to a big problem because the signals are not perfectly shaped like in solution. We have more or less like a big signal, so you can say that, because what we see is not just one orientation or the averaged out orientation. We see a lot of orientations.
And usually you want very thin signals so you can distinguish between different signals. But when they are all very broad, then they overlap too much and you don't basically see anything.
Yeah, that's true. So what I already said with with this, how the chemical shift comes up, I mentioned that it depends on the available electrons and the shielding and so on. So what does the chemical shift influence are also the electrons and stuff around. And when you have different orientations of your spin averaged over the whole sample, you have different interactions with the other surrounding electrons and stuff. So, and that's why we have a big, huge signal, and in the end, you see nothing. So this is you can read out things out of that. That's completely crazy. They are so clever people. They actually use those information, which is amazing. But for me, with macromolecules, I already have a lot of problems because I already have a lot of signals. If I would just see just this bubble things, so I could not see anything.
Yeah.
So we have to overcome this. And what they did, which they could not do it in the beginning, but they already calculated it, is two things. So at first we spin the sample because we don't have this small glass tube. We have a small rotor where our samples are inside, and we spin them. So if they are so slow, we help them and we spin them very fast. And we spin them at a specific angle. This is called the magic angle. And this is at 54.74 degrees. So just remember 54. And this comes from the Legendre polynomial. A very, very big equation.
So they calculated this angle before they could prove that it would work.
Yeah.
Wow,
that's exactly what they did.
Crazy.
So they did the math, and then they were like, okay, now engineers, please help us. Yeah, because it's like, I just say like, okay, we have our rotors and then we rotate it, but the engineering beauty behind it's crazy. It's because we rotate the rotor on a small air pillow.
How fast are those?
Depending on what you're doing. So, like in our lab, we usually go from 8 kilohertz up to, usually we do go up to 20 kilohertz.
Yeah, that's crazy. 20 kilohertz is like 20,000 rotations per second, right?
Yeah.
That's crazy.
But it's getting crazier. Now it's getting nerdy. So you have to rotate as fast as your interaction you want to overcome is, more or less. In solid state NMR, we do not detect hydrogens because the signals are too broad. With hydrogens, there's another problem. The amount of hydrogens in your NMR active hydrogens in your sample is super high. And what we do not average out to in solid state NMR is also the dipolar coupling. And in the dipolar coupling between spins of the same sort is very strong, and especially for hydrogens, because there are a lot of hydrogens. So couple very strongly. So what you see in the end is just a big something. It's like a hill. And
so no sharp peak, just
no.
a hill.
It's just it's just a hill. To be honest, it's just a hill. And then to overcome that, you have to rotate really, really fast, around 1,000 kilohertz. No, 100 kilohertz, but this is 100,000 hertz. So now it's correct. So this is pretty fast.
Damn, yeah. So like alone the engineering on how all these materials can withstand this extreme forces that are generated through this rotation is crazy.
Yeah. A thing why we rotate a bit slower is also because of the rotor size. You can't rotate big rotors with much sample, what you need, and to detect something. Because an NMR is not really sensitive, because this nettomagnetization, which I introduced in the beginning, this is just, when you remember it, just a few spins. So you need a lot of sample to actually see something. And for bio-NMR, you need a lot of sample because you have not a good resolution, as you have a lot of spins and a lot of, like, this whole molecule is super big.
No.
And therefore we can't rotate super fast, but it's okay for the things we detect, like carbon and nitrogen. The spinning frequencies, they are enough. But if you want to detect the hydrogen stuff, you have to go much faster, as I explained. There, the rotors they shrink, so they go from 4.2 millimeter to 0.3
Yeah.
millimeter.
Okay, that's tiny.
And imagine, like, the engineering part is crazy because in the beginning you had this with the angle, so you have to put in the sample and this has to be perfectly orientated in the probe pad, and then it has to rotate perfectly in the probe pad over a lot of temperature ranges where also things change, and then you go smaller and smaller and smaller, rotate faster and faster and faster, and yeah, it's crazy.
Yeah.
So we have those small rotors, and you fill them with like just drops of your sample, and we need a microscope for that. Yeah.
So to basically fill the solid in this tiny tube, you use a microscope?
Yeah, to see what you are doing.
Okay, interesting.
Yeah. Yeah, it's not, and then in the end, you don't know if it rotates.
Oh no.
Yeah, that's weird. Yeah, this is really crazy. So it's very expensive, but when it works, it's nice.
Yeah, that's a really unique way to measure it, because you can get a lot of information then that you wouldn't get otherwise.
What we see is that okay, what we do in the basic experiment in solid state NMR, it's called cross polarization. So here, because the 13C and 15N nuclei, they are not highly abundant usually, and also the gyromagnetic ratio is not super good. So what we do is to excite proton spins, and then we hold them under a specific condition, which is called Hartmann-Hahn condition. And here the magnetization goes from the excited protons to the unexcited carbons. This also has something to do with Boltzmann equation and warm spins and cold spins and stuff. So everything goes from warm to cold, and because that's how things work in thermodynamics. And now the carbons are excited, even if you did not excite them directly, which is nicely, because then the relaxation times are better than with the carbons. And so we can see the hydrogens indirectly because we can see which carbons we excited. And also,
I think for this is also important. Maybe some of the listeners don't know that only the carbon 13 is NMR active, and the probability to have carbon 13 instead of carbon 12 is very low. So that also reduces the amount of nuclei you can see, right? Or can you also excite with this cross... what was the name?
cross polarization. Yeah, maybe I mixed things up. I think it's a bit misleading. So the main reason why we do this cross polarization is that we uses the better gyromagnetic ratio of the hydrogens compared with the carbons. But we just see the 13C carbons, that for sure. And what we do we have to label our samples. So it's pretty expensive, but you can produce 13C isotopes via specific chemical experiments. So there are fabrics that do that. So industries,
Particle accelerator, I think.
Yeah. And sometimes also labs. So if you need something specifically, also some organic labs do that. And they label the things you want to investigate. And with the proteins, as we express our proteins in cells, we add the isotopes into our cell solution. And then we hope that E. coli bacteria.
So for your proteins, you actually increase the amount of carbon 13 in your sample to see more?
Yeah.
Okay.
Yeah. And also
Interesting.
nitrogen.
Okay, cool. And then, sorry, I interrupted. It's you
No.
use the bacteria to do that.
Yeah. So we have our bacteria. So depending on the things you want to investigate, you have to choose an appropriate, how is it called? Like bacteria or something. Sometimes people do things with yeasts. But in my case, thank you, God, I can just use E. coli because this is the easiest way to do it. And they do not stink that much as yeast.
Yeah.
Oh, it's crazy.
Yeah, one of the previous guests here also did some yeast research, and then she also said that is very smelly in the lab.
Oh, it's crazy. There are pictures of me with tissues in my nose because I can't do it. The smell is so crazy. Because one of my colleagues uses yeast.
So
Yeah,
good that
but
you can use the bacteria.
yeah, they are much better. And this is a very common routine. So this is the easiest way. And yeah, we just give them the isotopes and then they start to use those isotopes for their protein metabolism. And but if you want to label just specific amino acids, so in my case, I'm focusing on histidines and I just give them the histidines. Labeled. So you can choose fully labeled with 30C or 15N or just one or the other.
And then you get a lot more of these NMR active nuclei in your sample, and then that increases what you can see. Because I remember from the normal solution NMR where you don't do this labeling, the carbon 13 measurements would take a lot longer than the proton NMR because it's just so little active nuclei for that.
Yeah, exactly. So the amount of laterally active carbon nuclei is 1%. So we just have 1% 13C isotopes naturally, and for nitrogen it's worse. We have 0.4%. So this is not good.
So it's really important then to label it.
Yeah, we definitely have to increase the number for appropriate measurements because to run those magnets it's expensive, and also it's completely useless if you measure over weeks and the signal to noise is still shitty. So yes, that's why we do that usually, that we label our samples. And the second thing, the dipolar coupling, it's annoying when you just want to see something in the beginning, but sometimes it's useful if you want to check in solution NMR. You usually do those NOAs. So depending on the electronic interaction between the nuclei, you can say something about the vicinity of nuclei to each other, which gives you interesting information. And we can do the same with the dipolar coupling.
So you can derive the structure more easily because you can also see interactions between nuclei that are not necessarily next to each other, but just in the room they are next to each other, and they could be several amino acids apart in the chain, but in space they could be close, and then you can see those interactions.
No, not that easy, to be honest. In solid state NMR, the thing is you have to have a very precise question. Otherwise, you see absolutely nothing. But what we can do is to see if, for example, if there is a hydrogen bond between two amino acids, which are not directly next to each other. This is something we could detect before via other methods. But the hydrogen bond is something that we can detect with a dipolar coupling. And for example, if they are at another alpha helix, but it's in vicinity. But for that, you have to have the precise question because you have to label all those things. Otherwise, you see nothing, and you have to find the perfect experiment because dipolar coupling, to detect them, it's not easy because if you reintroduce them, so usually you rotate that you don't have them anymore. Now you want to reintroduce dipolar coupling. So there are specific pulse sequences where you reintroduce the dipolar coupling, but then you really have to know what you want to see. And that's something I do right now. And it's crazy because in the beginning, you don't know what you will see because you never did that before. And in the end, again, you just see signals, but you don't know, is this the correct one? What's that? Is it an artifact? So is it a real signal? What is that? Yeah, that's the thing.
Yeah, now you talked a bit about the pulse experiments. I think that maybe we should also mention that depending on these radio frequency pulse, how strong they are, how long they are, and how much time you wait in between, you get different results. Or how does that work?
Yeah. So the simplest experiment in solid state NMR or also in solution NMR is just to excite and then to detect. And depending on which, it's just a simple 1D spectrum. So depending on which channel you detect, you see the signals from the hydrogens or from the whatever. And the longer you excite during this first pulse, the stronger the excitation is. And yeah, now this is something one could say. So like you want to have the pulse as long as possible and as strong as possible to really excite everything. But the limitation is again the hardware. So you can't excite for every time. Usually for 1D measurements, we excite up to 5 microseconds because after that time, especially with the strength of the pulse, you get hardware problems. Probe heads are very sensitive. And for 2D spectra, so now we can see 1D, but we want to see some interactions. So to see that, we wait a bit. And then, depending on the pulse sequence and depending on what you want to see, you give in a next pulse. There are a lot of things you can change. So like the length of the pulse or the direction of the pulse and how much you excite the signal. So we differentiate between 90 degrees or 180 degrees. And depending on that, you see different phenomena. And if you go back to this coordination system, in the beginning, we excite from Z to the XY plane. Then the spins go back. And if you excite them again, they are not completely back in the B0 along Z. So you find them somewhere in between. And this gives you information because here they have, and again, another kind of chemical shift. And this gives you cross signals, and there you can get a lot of information.
So there you could detect something when you excite the protons, which are easy to detect, to say it simplistic. And then with that, you basically look at the interactions of the protons with the carbons. And then you can get information about the carbons without having to excite the carbons itself.
Yeah, more or less. You can do that, but you have like the pulse sequence is a bit different. But in general, it's like that. Depending on where your pulse is, on which channel, you can see the difference. Yes, yeah. Pulse sequences are a big thing for them.
Could be an episode of its own.
Yeah, definitely. But the easiest way is to excite the hydrogens twice.
Oh, okay.
So that's the easiest experiment, and this is very useful too. Yeah, this is actually something biological NMR people from solution state NMR do to assign the amino acids of a protein and in the end to resolve the structure. So now usually we use X-ray or electronic microscopy, but solution NMR or NMR in general is still useful especially if you want to have the dynamics. That's a big point of NMR. Dynamics, they are still existing in NMR, so we really can see what happens on a dynamically timescale.
And when you now come to your solid state NMR, what is the advantage to see the proteins in a solid form instead of liquid?
So we don't look into soluble proteins for sure, so they are perfectly in solution. No, we want to, like I do, to investigate membrane proteins. And yeah, membrane proteins are super important to investigate more specifically, because the cell membrane is super interesting, what happens there. It's also, if you, like, one of my colleagues is working with LGBT2 BFG. Yes, I think I put it in the correct direction. And this is a protein that brings LPS in bacteria cells in gram-negative.
What is LPS?
LPS is lipopolysaccharide, so it's something important for the gram-negative bacteria. They put it outside of the like on the outer membrane of them, and they have a structure built by those polysaccharides, and this structure makes it really difficult to kill those bacteria. So, like, it's their shield. Yeah, I think that's a good thing.
And
it's a shield.
With the solid state NMR, you can look at those, which would not be possible because they're not really soluble.
No, we can look further into those LPS protein moving proteins. So this LPTB2FG, this is the protein, or it's part of a big protein that transports the LPS from the inner membrane to the outer membrane, and you can't really see how it functions, this protein, when you just look it into solution, because it has to be in the membrane to really see how it functions. So it has to be in the most native surrounding, and that's the biggest point. So we have membrane proteins for sure. In detergents or in nanodiscs, we can investigate them, but it's not their native environment. So the information we gain there is maybe not correct because it's not in their native environment. So the best way to investigate membrane proteins is for sure to investigate them in membranes. And membranes are solid, and that's why we use solid state NMR.
Okay, so you don't have to modify them. So with this solid state NMR, you can look at them how they're actually, or close to how they would function, and not in solution where they get denaturated.
Yeah, no, they're not denaturated, but in detergent. So yeah, they are in those mice cell things. So yeah.
Okay.
So now, if you like, this is a good example. This LPTB2FG. Oh my god, this is so long word name. It's a big nice example in general, like why this is important, because if a lot of gram-negative bacteria are resistant against antibiotics, and this is a big problem. And this fundamental research that my colleagues and a lot of other people from my university do is to investigate those proteins and to help how they function, and to help to find solutions how we can overcome them, or this LPS shield barrier, to overcome those resistances, because multi-resistant bacteria, they are not very helpful.
Yeah, that's a huge challenge. Okay, maybe we can now go to your research. What do you use the solid state NMR for?
Yeah, so I investigate xenorhodopsin. It's a microbial rhodopsin. So rhodopsins are photoproteins. Nice thing is they have a color because they absorb light and imitate light. So then you see a color. And like there are a lot of reasons why those proteins do this. In our eyes, it's not a microbial rhodopsin, but we have also rhodopsins in our eyes. So we use this phenomenon of light-induced reactions to see something. And
These rhodopsins
but
do similar stuff, but not in the eye.
Yeah, that's true. So like also like more or less in like photosynthesis is also kind of a photoreaction.
Yeah.
Or it's definitely a photoreaction, but yeah, not with the same thing. But microbial rhodopsins, there are a lot of organisms, so you find them actually everywhere, and it's a way to produce energy or to use the energy from the sun. And there are a lot of functions, so some of them pump the ions, some of them are like there's an activation of a ion channel, or like a sensor of something. So there are a lot of functions which are useful for the organism they originally come from. And my xenorhodopsin, it's from Nanosalina, and it's a archaea, so it's not a bacteria and it's not a parasitic cell. It's something else. To be honest, I don't really know it because I am not a biologist.
Very primitive life forms. I think they're much older even than bacteria.
Yeah, definitely. And they live in very weird environments. So this Nanosalina xenorhodopsin comes from Lake Tyrrell in Australia. It's a salt lake. And this is a proton pump. And usually the proton pumps we know or we knew are outwardly directed proton pumps. So they pump protons from the inner part of the cell outwardly directed. And this xenorhodopsin is inwardly directed, which is pretty nice because now I'm doing the fundamental research stuff to understand how the photocycle, so this is called this whole process where the protein uses the energy to do the proton pumping, the sun energy. But in the end, where all those information end is called, this field is called optogenetics. And optogenetics is something that you can imagine that the aim is that you can activate processes with light in animals. So they use those proteins already in rats and other test animals to influence ion flow. So this is important in for example in brains. So this is very important to understand neuronal networks and how information goes from there to there. This is all about ions in the cell or not, how the information goes around.
Interesting.
So this is this is the aim. But I'm doing the fundamental research. So xenorhodopsin, like my favorite purple protein, this has this like, with solid state NMR, what can I do? So maybe I start with that. So we work on the dynamics. Again, this is the big advantage of NMR in general, that you can see dynamics. And what we do is to see how specific important amino acids behave during the photocycle. So how are they orientated? What happens when are they getting protonated? That's something we can see.
So you basically look at the reaction while it's happening.
Yeah, more or less.
Yeah, cool.
That's the thing. Again, we can see, can we excite by cross polarization our spin or like our atoms. So if there is for example, my favorite amino acids are histidines, and histidines have this imidazole ring with 2 nitrogens. And they can do a lot of things with, they can be protonated or not, they can be in a neutral and not in proteins, but in general, they can be also like completely deprotonated. And when they're getting protonated, we have a much better signal at a specific chemical shift. And when they're getting deprotonated, we see them at a different chemical shift, which is pretty useless as I have a proton pump. So I can see if maybe the proton comes along this one specific histidine during the photocycle. So is it getting protonated? When it gets protonated, chemical shift change and the CP transfer is much better because we can actually go with this Hartmann-Hahn condition. We actually have this cross-polarization step because we have a directly connected nitrogen to proton.
Cool.
Yeah, that's pretty cool. And so that's what I'm doing right now. I investigate on a functional basis which amino acids are important, what are doing my histidines. And the other part of the project is more structural. So I did a lot of mutations, and I just saw what happens with the important amino acids.
So you change out these amino acids in the proteins, and then you see which one of them are crucial for the mechanism or which ones are just there.
Yeah, and we can also see how they influence each other. Yeah. So if you mutate something, you can see how much does the chemical shift change, how much does the shape of the signals change, and because this gives you a lot of information what happens. So like when you change the environment of a spin, for sure the chemical shift changes. But sometimes you change something at one end of the protein, but this has an influence to the chemical shift of a completely far away other amino acid. And now things are getting interested. And this is something we can see.
Then they might be close to each other in the structure, or is it just other influences?
Yeah. So we usually we start with our X-ray structure. And so when I mean far away, I actually mean far away. So they have nothing to do with each other. But you never know how they are connected to each other. Is there a hydrogen bond network or whatever?
And also how the protein folds, I think also plays a role, or?
Yes, that's true. So in general, if it does not fold, you definitely see it because you have no protein to detect but what one can also see is if it folds differently. Sometimes you have a pentameric state, which is the common way to that this protein oligomerize, but sometimes you change one amino acid and then you end up with a trimer. Yeah, proteins are wild.
Yeah, that's I only did the chemistry and used NMR for like really small in comparison molecules with like 10 maybe carbons, and that's then a completely different world. So for me, it's really amazing how you can derive anything from these huge molecules with the NMR. That's really interesting.
Yeah. So I think the bigger the molecule gets, the more precise your questions have to be. Yeah. So the thing is, you can't measure everything at once. And then, that's why I just focused in the beginning on histidines and how they behave during the photocycle. And now things are getting more complicated, but I started with that, saw interesting effects, and then I was wondering why are they happening, and then the next question came after and after and after.
Yeah.
Just a short note. I said that I investigate the photocycle with NMR, and this sounds pretty easy when I say it like that, but actually it's not super easy. But it's not because it's super hard in general. because this is the thing this proton does. So it has this photocycle. But again, we have a lot of like hardware things we have to consider, and that's the part. Like solid state NMR is a lot of hardware stuff.
Get light into there is probably difficult.
Yeah, like at first you have to have to try to put light into your probe head. Yes, and here we use something which is a mixture of EPR and NMR. So what I use to investigate the photocycle is it's called DNP, dynamic nuclear polarization spectroscopy, and this is just a mixture of EPR and NMR. You use radicals to in the end what you want to have is a signal enhancement, and there you use the radical, and due to a lot of Boltzmann stuff things, you actually change this netto magnetization. So as I said in the beginning, you had just a few spins you can see their excitation, and with dynamic nuclear polarization you change this population, so you have a lot more spins you see in the end in your spectrum. That's why you have the signal enhancement.
So these radicals somehow change the ratio between these two levels, and then you can excite more of the spins and see more.
Those radicals are electrons, so you excite the electrons, and then you have you have an effect, this polarization effect from the electrons, excited electrons, to your proton spins, and here that's why you change the Boltzmann distribution, because you have this polarization. Yeah, and this is, for me, it's super nice because I have a signal enhancement. And as we measure at 100 Kelvin, we can trap the photointermediates. So we start at 100 Kelvin and illuminate our sample. And then we start to increase the temperature a bit and lower the barrier between the photointermediate states, the energy barrier, and then it hops from one a photointermediate to the next one. Then I measure again, and then I go up again, go from the next level to the other one, and so I can like trap all the intermediates along the photocycle. And in my case, I'm most interested into the histidines.
Do you have to take out your sample every time, or did you manage to get the light source in the spectrometer?
No, we have a light source in the spectrometer. So we have a probe head with basically a hole.
Okay.
And there you can put in a light channel, but you just illuminate in the beginning. So you illuminate to excite everything from the ground state to in this case, it's the K state. That's the typical name. So the first photo intermediate, and then you can stop the illumination. And then it's just thermodynamics. And then it goes, and in the end, you end in the ground state. And here I can really nicely see what happens with the histidines during this photocycle or other interesting amino acids. So this is a really nice and very important technique for microbial rhodopsins in NMR.
Interesting. It's a very specific field and also fundamental research, so I think it's sometimes a bit difficult to derive what future stuff might come from it, but also it's like with the NMR, at first they figured out what magnetic resonance is, and then it took decades until NMR was basically usable like we do today. That's why the fundamental research is so important.
Yeah, and it's again like it's a thing with fundamental research. You do things, and you don't just get money, so you have to explain the DFG or whoever that the science you do is important, and they see, okay, seems to be important. Then you do the science, but in the end, with fundamental research, even if your idea was nice, maybe nobody will use it.
Yeah.
So that's the thing.
Yeah. You never know what will come out of it. But that's also the pure science, I would say. That's what we have also discussed in one of the previous episodes, where you really do it out of curiosity and you just want to figure out how stuff works without the goal of making money, which is what the research in the industry does.
Yeah, definitely. And I mean, those inwardly directed pumps, to understand how they work, and especially with the application of optogenetics, it's very interesting because this is a huge field. And I'm pretty sure that for optogenetics, there will be a Nobel Prize in the next decade. Definitely. It's so interesting, the things they do, and I'm pretty sure. And so we will see what happens. And in general, it's nice to know how proteins work, because you can transfer things you saw somewhere to other things you need right now. And that's how science works. So you, in the end, if you are a scientist, and yeah, you just check out what do we already know, and how can I apply this on my research field. And in the end, to know how proteins work is pretty good, because there are a lot of proteins, especially also like humans. All every time,
Yeah,
just think about humans, what is important for
yeah,
humans, but we also have a lot of
of
proteins.
course.
And yeah, I'm pretty sure that nothing is for zero reason, but
Yeah, there will always come out something, because you just contribute to the overall knowledge and the toolbox that others can apply. And then it's very rarely that one scientist does everything. And especially the more advanced we get, the more we need to do this fundamental research, that we basically can find out new stuff for others to work on.
yeah. So all the fancy things now, they are based on fundamental research.
Exactly. And when it comes to the NMR, do you see there any new advancements that are maybe coming in the next years or in the upcoming future?
Like for solid state NMR, as I mentioned frequently, we really have a hardware
Yeah.
problem. So not a problem. So it's really nice right now, but I think in hardware cases there is a lot to improve. So starting with those pro pads. So right now, some of them are very sensitive to temperature changes, and they can't translate the different radiations homogeneously to the whole sample. So maybe we will find a way to really excite the whole sample, not just parts, or I don't know. There are so many things from hardware sides, from pulse sequence sides, which is more interesting for the science. There's already a lot of things that happened, and that that are already discovered. But there are also again things which is a bit connected between the hardware and the pulse sequence. It's called optimal control. Yeah, basically you try to find the best way to pulse in the B1 field, so the radio frequency. And this is in a way a mathematical approach, but this is also limited by hardware. So I think hardware.
When it comes to the analysis of the spectra you get, do you think there are AI tools coming up? Because I think especially when you have these really difficult to interpret results, it might be, yeah, very useful to train AI on that and then get better results just from that.
Yeah, we already have that. So we already have signal assignment AI software. They are okay. So for especially for soluble proteins or soluble substances, for membrane proteins, or in general everything in solids, it's not as easy. They give you a first hint or a first tip, but you definitely have to recheck that, and it's not every time super correct.
Yeah.
And yeah, but they are very useful when you want to have a overview.
Yeah. I think there will be with when you imagine how fast all these AI tools are moving compared to like only last year or 2 years ago to compared to what they can do now. So that's already crazy how fast this moves. So I hope that this also will get into all these chemical research and then help to accelerate all this research.
Yeah. So at last conference I have been, there were already some industry parties that introduced their software, and in my university there were a professor that already worked on signal assignment of NMR protein signals, which is bit difficult. Yeah. Yeah, proteins are more or less living things, so they have dynamics,
Yeah.
and this changes the chemical shifts. But in the end, it's better than nothing. So yeah, so there are things. AI is super useful.
It's very interdisciplinary that you have. In this case, you would need probably physicists for the NMR or engineers, and then the chemists and biology is also in there, and then you would also need computational chemists and then some AI researchers, and they all would have to work together to really make a huge impact, I would say.
Yeah.
The more advanced we get in this, we have to collaborate with different fields.
Definitely. Already, like my research group, we are like, my boss is a physicist. This sounds so weirdly in English. And like, I'm a chemist, my colleagues are biologists or biochemists, and we are doing an analysis method where you can come from a very physical point of view or just do bio things. And yeah, that's why we are called the Biophysical Institute. So there's a lot of things one can do.
Yeah, interesting. And then also what you do is talking about all kinds of chemistry in your podcast you also have. What keeps you motivated to do also this podcast you have?
I think I like to go deeper into things I think are interesting. So, like, at one time I was wondering, like, okay, what caffeine really does to me, and then I just like thought, okay, maybe this is an interesting episode for my podcast. So I started a deep dive. Like the last episode, which I published yesterday, or when you hear, I don't know when you hear that, but in the middle of July, is about microbiome and the brain connection, how everything is connected to each other. And yeah, and I just like to read those things, to go deeper in those things, and then to write it down in a hopefully simple way for everyone that listens. And then, as I like podcasts, I think that's the starting point for every podcaster. We like podcasts. And I just started with this for years ago, and yeah, I still doing it, and I really like it, and that's why I'm doing it. It's just intrinsic interests, and then I saw that people like it, and then yeah, that's a good motivation, I guess.
Yeah, definitely. This talking about science, I think, is in general important to also get other people more interested. And I think with your podcast, because it's on a very easy to understand level, it can also be for someone who is not really an expert in chemistry, and they can listen to it and yeah, maybe start studying chemistry because of it.
Yeah, or maybe start to see how things are connected to each other. So when I do the podcast, I start to go deeper into stuff, and then sometimes I end in a completely different direction because everything is connected to each other, and that's something I also show them. So I make connections to recent studies or what I am doing. So everything is connected and the world is connected, and that's helps maybe people to see how the world works and helps them to see, okay, interesting. Now I know more.
Exactly.
Yeah.
That's why chemistry is also so fascinating for me. That it's everywhere, and you can explain so much. That when you just look at it, a lot of stuff can be explained by the chemistry behind it.
Yeah, definitely. Chemistry is really fascinating. That's true.
So last episode, I started this closing tradition on this podcast is the question, what is your favorite discovery in chemistry?
Yeah. In chemistry. Oh, I thought in science in general.
Yeah, we can also do in science in general, I guess.
Okay, perfect. But because I really thought about this long, like it was not so easy for me. But what really fascinates me since I'm a child is that we can fly into the space and general what we already know. So in space there are also spectrometers, not NMR spectrometers, but with optical techniques, and maybe this is also kind of chemistry, but this the science which is which is currently happening in space is also really interesting, and this just really fascinates me, space stuff, space science.
Yeah, definitely. Space is super fascinating. I really like this James Webb telescope and what they can all now show in comparison to Hubble, and it's truly fascinating.
Yeah, I'm so nerdy during Corona. So like the first month, I did a Coursera course, astronomy, exploring space and time. So this was what I did during COVID.
That's a very productive way of spending that time.
It was so interesting. And they explained how I can really recommend that course. It's for free on Coursera. And they explained also this Higgs particles and stuff.
The God particle, right?
Yeah, it's really interesting. So
yeah,
I can recommend that.
Yeah, definitely space is a super interesting topic in science. There's also so much more that we can discover.
Yeah.
Yeah, with this, I think we're coming to an end of this episode. Thank you very much, Anne, for this discussion. I learned a lot about solid state NMR.
Thank you very much for your invitation.
Thank you very much for listening. Please consider following on whatever app you like. This really helps to reach more people and you won't miss any episodes. Also, check out Anne's podcast if you speak German. It's called Darf's ein bisschen Chemie sein. I'll put a link to that in the show note, and also the books Anne mentioned if you want to dig deeper into NMR. Until next time, keep innovating and stay curious.